Good afternoon, everybody. Thank you for being present here. So my name is Rutger van Diest. I'm the Global Head of Sales for the CDMO activities within Bavarian Nordic. And I will give this 30-minute presentation where we'll discuss the different attributes that we took into account for establishing or setting up our complete new fill and finish facility in Quistgaard, where our headquarters are in Denmark, to enable us to work with live viral vaccines. So through the presentation, I will first give you a little bit of information about Bavarian Nordic, who we are, what we do, where we are located. Very short intro about the vaccine market. What is the size of the market? Who are the big players? How are the trends for the next coming 10 years? Introduction about live attenuated viral vaccines. What is specific about them? Why are they so complex to work with? And then link to those complexity, how we did the basic design of our new facility. So Bavarian Nordic, we are a global vertically integrated biotech company focused on bringing vaccines to market. So we do that by development, manufacturing and commercialization of vaccines with a focus on infectious diseases and cancer immunotherapy. Our goal is to improve the health and quality of life for children and adults, focusing on indications for which the unmet medical need is high and for which we can harness the power of the immune system to induce response. So we do have a few business pillars, I would say. We have our commercial platform, where we have the smallpox vaccine. We also acquired recently 2 vaccines from GSK, so Encepur and Rabipur. We also now have our own pipeline where we focus on infectious diseases, where we have also big collaborations together with the big pharma, and we also have our immunotherapy pipeline. Besides that, we started beginning of this year actually to open up the facility for CDMO activities, so that we can always fully help our customers with drug substance or formulation development, drug product, also in the new facility that will be ready as from next year. So we know we're not a new kid on the block, so we're already 25 years on the market. So everything started about 25 years ago with some Danish scientists based in Copenhagen and some scientists in Germany, in Munich, hence the name Bavarian Nordic, when they came together. We have around 490 employees worldwide. So we have 3 facilities, as you can see. Headquarters are in the middle in Quistgaard, which is about 30 minutes north of Copenhagen. There we have the multi-product manufacturing facility. We have QA, QC over there. In Germany, we have an R&D facility located in the Munich area. And in the US, we have clinical development and regulatory affairs in the RTP triangle area. Which is important is that we are also a scientific-driven company, so more than 40% of our employees hold a PhD, so meaning that they are really experts in their field of expertise. We are listed on NASDAQ, and the first product we had approved in the market was in 2013, was our smallpox vaccine. It was approved for Europe and Canada at that time. We supplied it also already to the US as an emergency drug, but recently, about a month ago, it was also approved by the FDA to be used in the US. And an additional bonus, I would say, is that it was also approved for a vaccine against monkeypox. As said, we do have long-term R&D and delivery contracts with the US government and development partnerships with industry partners. What we offer, actually, we work in the complete value stream of bringing vaccines to market. So we start from development, process development. We have experience with tech transfer. We have the master seed virus that we then build up in our German facility. It will be brought afterwards to our facility in Denmark, where we do bulk drug substance manufacturing. We have then, currently in our current setup, we have a small scale filling setup, so we can go up to batch sizes up to 10,000 units, but we are building now a new one, and you will see it later on, that will be operational as from next year, and that's really into large scale manufacturing for vials, and everything there is done with Isolator technology and lyophilization. So that's a little bit in a nutshell BN. The vaccine market, so I show you some numbers from 2018 and a trend towards 25. So you see last year total market in the vaccines was around 34 billion US dollars. It will go up to almost 51 or above 51 billion US dollars in 25, which is still, it's a big market share, but if you compare that to the pharmaceutical industry, it's still a relatively small market. But we see much more and more uptake by the pharma into the vaccine business, and that is because the high prevalence of infectious diseases. We see more and more government support for R&D into the vaccine manufacturing, launch of new therapeutic vaccines. However, we all know that it requires a huge capital investment for developing vaccines, and that may restrain the market growth. One of the major trends we see over the next 10 years is that vaccine suppliers will rely more and more on outsourced manufacturing to companies that can offer a specialized service into their field of activities. If you look at the market share, North America is the highest market share. It's around 14 billion versus 12.4 billion versus Europe, so still the biggest, but the gap is small. Also important to know is that around 80% of all global vaccine sales comes from the top 4 companies. So they are representing actually 80% of the total market sales. So that's Pfizer, Sanofi, Merck, and GlaxoSmithKline. And then you have the other companies representing the remaining 20%. From a vaccine type, so I would say the dominant vaccine type is the conjugate vaccine, 40% above, then followed by the live attenuated vaccines. Those are the ones that we focus on within Bavarian Nordic, and they're more or less the same size as the recombinant vaccines. Pneumococcal diseases is the #1 indication, followed by influenza, and then you have a few around 10%, and then it goes down. Patient type, obvious, pediatric is still the majority of the market, and monovalent is above 50% of the valence type versus multivalent. And then route of administration, you see the majority today is intramuscular and subcutaneous administration. Live attenuated vaccines, how we do it at Bavarian Nordic. So this is a typical, I would say, process flow of what we are doing with our own vaccines. So we start with the master virus bank. It's created in our German office. It's brought down to Denmark, where we can then have the working seed virus to inoculate our cells. For the bulk drug substance, we have a large experience with CEF cells, so we still work with the egg incubation, we do CEF preparation, and then we have 3 different types of technologies with the CEF cells, so we can or work with the roller bottles, wave bioreactors, or with the single-use bioreactors. So it means that the cells are inoculated with the virus, then we use a technology whether these roller bottles or wave bioreactors, and we do the purification step. Cell cultivation, so it can be done with our CEF cells, or we could also use, for instance, other types of cell lines, Vero cells, or anything that can be worked out in the bioreactor. So you see a picture of the facility in Denmark that we use for drug substance and small-scale clinical material and manufacturing. The drug product, typical process flow is you start with wash and sterilization of your utensils, then you start with the formulation of the product, filling. In case you have a lyophilized product, you go to your lyophilization step, capping and external washing in case it is a BSL-2 or a live attenuated vaccine, visual inspection, labeling, packaging, and you put it into the warehouse. So for that activity here, we're setting up a new site. You see a rendering, so the complete building is already set up. All the equipment is in place. We're doing IQ/OQ testing, and next year we will do qualification to have new products introduced as from 21. You see our filling line. I will come back on that in detail. So it's a filling line from Bosch, and we have a freeze dryer coming from EMA. So everything is designed to be GMO2 or BSL-2 classified. Everything or every product contact is single use, and we work a lot with Isolator technology. So what is the complexity driven by live attenuated vaccines? There are 2 ways. So it's the nature of the product that makes it complex for us to handle these products, and it's the required environment. So the nature of the product, we know these are live attenuated viruses, so we need to have specific precautions of that. We know these are very complex and fragile molecules. We need to have processes in place to make sure we don't break the molecules, that we have temperature protection, and it's also expensive products. From an environment to manufacture and to handle the products, we need aseptic manufacturing. There should be the required biosafety level, and of course we would like to have a multi-product facility to have quick changeovers. So it's resulting all in a complex manufacturing and supply chain effort and process. for which a high level of expertise is required. If you go into detail, so the nature of the product, so live attenuated viruses, into our design we took into account that to make sure we can handle these products, we need adequate biosafety level, adequate cleaning and decontamination. It's a full single-use technology setup, and we have full isolator technology. To handle the molecules, we go for high-speed filling lines to make sure that the time out of fridge is as short as possible. We have low-pressure peristaltic pumps to not break the molecules, lyophilization capabilities for stability, minimized transport between activities, cold storage, and of course a reduced time of refrigerator. The fact that these are expensive products, also we would like to minimize, or we do all efforts possible to minimize product losses, and we enable small bulk volumes. So the basic design, now knowing that what the complexities are from the products and the environment. So our goal is actually to have a multi-product site available for fill and finish activities and lyophilizations of live viral vaccines and more. So the basic design requirements are we need a multi-product facility with biosafety level 1 and 2 ready to go a step up into level 3. Of course, we have aseptic manufacturing, so we will discuss fill and finish operations, flexible packaging and serialization, and of course the adequate warehousing and storage. And then of course we need to be in compliance with all the authorities and with the CGMP guidelines. So if you talk about the multi-product facility with the biosafety level, now, excuse me, the BSL level, so biosafety, what is it actually? So biosafety is the application of safety precautions that reduce a laboratorian's risk of exposure of a potentially infectious microbe, and limit contamination of the work environment, and ultimate, of course, then further down the complete community. What are biosafety levels? You can see it here. So there are 4 different safety levels, biosafety levels. And the primary risk for each of them, or how to determine one of the biosafety levels, is dedicated or defined by infectivity of the microbes, the severity of the disease, transmissibility, and the nature of the work conducted. Of course, also the origin of the microbe and the exposure, the route of exposure is also important in that. Each of those 4 biosafety levels, they have their own specific containment guidelines. So we talk about laboratory practices, safety equipment, and facility construction. And if you go to the website of the Centers for Disease Control and Prevention, this definition comes out of that. And they also give you a very brief overview of what is needed to get specific classifications. I will not go in detail, but you can see we have in Bavarian Nordic BSL2, there are a few things that are ready to implement already for BSL3, but for instance, BSL3, to be classified, you need your laboratorians to be immunized already for the product they are working for. So the design of the building, so you see here a rendering of the building. It contains 3 different levels. We have the manufacturing level in the middle. On top you have the HVAC systems, and on the level 0 you have the utilities. And actually this is then the floor map, I would say. So we will have here, we have our, I can show it maybe on the next slide. So you see the different steps as we said from the beginning. So we have our washing and sterilization area where we do the washing and sterilization of the utensils. We have vial washing, vial dip organization, and the vial filling. This all is in the isolator. If it's a lyophilized product, we can go to the freeze dryer and we can bring it down to the vial stopper and capping. Afterwards, we can go to the storage area, which is in between inspection and packaging. It can go to inspection, packaging, and it can go out through specific killing autoclaves. If it is a diluent, for instance, we can pass it after filling through a terminal sterilization autoclave. So aseptic manufacturing. So here the basic steps with the equipment we have in-house. You see here we start from wash and sterilization. We went through it in the beginning, so up to the warehouse. And if you talk about the filling and the fill and finish operations, we have here the 5 big steps in the process. And we will now go over each of them, and I will explain you specific attributes we brought into place to make sure we can work with live vaccines. So wash and sterilization, we have utensils washer, the autoclaves, and the terminal autoclaves. We have The equipment itself is from Getinge, so we have utensils, washers, autoclaves, and terminal sterilizers. Volumes ranging from 810 liters to the terminal sterilization up to 4,000 liters. That's a big one with 14 shelves where we can bring in the filled product. So this will help us to reach the adequate biosafety levels. It will also help us to have the adequate cleaning and decontamination and the inactivation after processing the products. From a formulation perspective, we work with a set initially with a complete disposable setup, so single use. So we have a mobile formulation vessel from Merck Millipore. It's a 200 liter standard today. The tank as such can be temperature controlled up to 40 degrees. And then of course we have the floor scale and table scale. And as you can see, it's a single use setup. There is no open product contact, so within the facility everything is closed. If we need to add excipients, for instance, we will prepare them in an adjacent area and then bring them to the formulation area, and with sterile connections connect them to the formulation. And as said, yeah, it's temperature controlled. The filling process, you see a rendering of our process. As said, everything is except for the freeze dryer is from Bosch. So you see here the washing line, the tunnel, filling line, stopper capping line, and here the isolator up to the conveyor to the freeze dryer, which is from IMA. So we have chosen for a Bosch standard washer because of the high reliability and the proven technology. We also have the newest generation of tunnels coming from Bosch with a sterilizable cooling zone, glass partition walls, and light inside the tunnel to verify, for instance, glass particles eventually, and standard pressure cascade. We have a 100% in-process control at the filling line. We have, before we do the capping, an inspection 100% to see whether there is a raised stopper that could give a contamination or could lead to it, and a 100% after crimping to make sure everything is crimped within specification. And then to comply with the BSL2 requirements, we have an outside vial washer. If you have all these different process steps differently, so the Bosch filling line with the isolator, so the one we have is capable of working or running 2 ml vials up to 20 ml. It's a high-speed filling line, so the normal speed is 400 vials per hour if you talk about 2 ml vials. There's inspection, visual inspection for empty glass. We have 8 filling needles, 100% IPC. As said in the beginning, we have peristaltic pumps to make sure that we don't break our molecules. Filling volume can range from 0.1 ml up to 20 ml. Single-use filling equipment, and the crimping machine is at the end of the filling line, and that's in an so the complete line is in the isolator. The crimping machine is a closed wrap system. For us, important high speed. We need a high-speed line to shorten the time that the product is out of the refrigerator or out of a temperature-controlled environment. We want full isolator technology for inactivation and decontamination perspectives, peristaltic pumps, single-use setup to avoid cleaning steps, so we can make sure everything is inactivated afterwards, the vial washer, and then of course, because we have expensive product, we want to minimize product losses, and that is part of the Bosch line where we have the dose in, dose out unit to make sure that we can fill to the end of the bulk and avoid foaming at the end of the tubing. The freeze dryer, it's a large-scale freeze dryer from IMA. It is in an isolator from Bosch environment, so the size of it is 40 square meters. We have 15 shelves, and that allows us to have a full batch size, if you talk about 2 ml vials, of 180 vials per batch. Automated loading and unloading, and then of course, yeah, everything inside, the cleaning is done with SIP to make sure everything is done in an automated way. Important is it's also a high-speed line, so a high-speed loading, so it can follow the filling line with 400 vials a minute. It is in the full isolator technology. It means also that before we bring in the vials inside the isolator, the shelves are already pre-cooled or pre-frozen. Relatively humidity will go down in the isolator, and then thanks to the size of the freeze dryer, we can have a range from small scale ranging from 12,000 vials up to 180,000 vials. So in one of the design requirements, we said we want to have an isolator protection. So this part, the filling and the freeze drying, is done in an isolator. Capping and external washing is done in a closed wrap protection immediately following after the isolator. So the isolator, again, it's a Bosch one. So the areas or the processes protected are the filling, loading, unloading, the freeze dryer, and the capping. It's grade A. We have 3 separate isolator areas, so filling, the crimping, and loading. We have VHP cycles for decontamination of the isolator before we do a batch, and after a batch we can use the VHP cycle for inactivation. Important for us, it's a multi-product, so the isolator, for instance, if we have one product running for 5 days in the freeze dryer, we still can add another product on the filling line. Adequate biosafety level and decontamination and inactivation thanks to the isolator and the VHP cycles. We can reduce relative humidity after freeze drying, product and operator protection, and of course there's minimized transport steps between the different activities. So this is, for instance, the isolator part after the depressurization tunnel. Next one, you see the isolator that goes to the freeze dryer. And it means, like I said earlier, if you're now working, if you have a product in the freeze dryer, we still can run the line for a liquid product. This is then the end for the crimping of the caps. Visual inspection. So there we work with a semi-automated inspection line. We do have room and plans to go to a full automated inspection. For the time being, for our needs, a semi-automated inspection line is good enough. That comes from Sidenander, and it can be used for inspecting liquids, so both suspensions and solutions, and also freeze-dried products. From a flexible packaging and sterilization, so after the products are filled, we go then to the labeling and the packaging line. Here again, this is the high-speed labeling and packaging line we have from Bosch. So it will follow the same speed as the previous activity, so 400 vials a minute. Batch size, the vial sizes, the line can be adapted to work with 2 ml vials up to 20 ml. Labeling, the carton erecton unit is done, and of course sterilization. I will come back on that to the next slide. For us, it's important that it's a flexible packaging line so that we can work with small batch sizes, larger batch sizes, also depending on the countries that we distribute to. Product protection, so we can have the labeling, for instance, for anti-counterfeiting, and it's a high-speed packaging line in line with the other activities. Sterilization and aggregation, so we go to the individual numbering of the smallest sealable unit. In our case, for this product, it's a box, a carton or cardboard box with 20 units. And aggregation, we do this then on the case level, where we put the boxes in a bigger cardboard box and a shipper box, and then we go up to the level of a pallet, and then we can trace back what is on the pallet, how many boxes, and which individual units in each shipping box. To be able to link our system for the sterilization, so to get the numbering, we are quite flexible on how we can interact with our customers. We can say, okay, we take care of the serial numbers, we take the ones supplied by our customers, or we find each other somewhere in the middle where we have a system that we work with a cloud-based system to make sure everybody get the right information on time. Warehousing and storage. So there again, I'll show you within the facility. So we have quite a lot of storage capabilities and storage place in the facility, also externally. So what we have is, of course, storage for room temperature, freezers where we can put in the products that required temperature, a lot of 2 to 8 degrees. And then there's also quite a lot of -20 and -50 degrees also for our own products. So we will minimize the transport between activities to make sure that, we know how costful it is and risky if you take products out of the fridge. It's called storage capabilities for different temperatures, so whatever the product needs, we have an option to store it in-house. The site as such, it is designed to be in compliance with the EU and USA guidelines. The initial design was done in 2017. In May or March 2018, we had a Type C review meeting with the FDA, and we did get some useful recommendations. We implemented them in the final design, and we don't expect any issues to be in compliance with both EMA and the USA at US FDA. Of course, as said earlier, we have now also acquired 2 new vaccines. We will also distribute them, so they will be distributed to a whole lot of countries, and we have to make sure, of course, that we are then according to those regulations, that we are in line with them. Or if you have customers with a specific request for specific countries or regions, we can work on that. So the timeline, so everything started in Q2 2017. As said, the FDA Type C meeting was in 2018, and we started construction beginning of this year. Today, installation is done of the equipment, and we are now working on SAT, IQ, and then to start as from 2020 with the qualification, so PQ and tech transfer, and then by end of next year, beginning of 21, we do the handover for commercial filling of own products or customer products. So as a conclusion, the design of the facility is GMO2 and BSL2 classified, so we took all the recommendations and requirements into account. We defended them to the ministry in Denmark, and they approved the classification. And so we can say that the facility allows today for fill and finish activities for live viral vaccines within the technologies that we have available. So it means that also if you want to bring in a new viral vaccine within the facility, we have to perform a risk assessment. We will have to do the risk assessment on containment level. So is it BSL 1, 2, 3? And that is really country specific. So we have a customer and their product is BSL 3 classified in a specific country. If we do the manufacturing in Denmark, we were able to downsize that to a BSL 2 classification, because you have to be in compliance with your local guidelines. And then of course, yeah, we do a final risk evaluation of the specific version of the virus, and then to make sure that we can take everything in-house. That was an overview of what we are doing, why there are so many there is complexity in handling live viral vaccines, what we took into account to make sure we can do this in our Denmark facility, and the timelines.